you're ready. Yeah, thank you.
Thank you so much for such a kind introduction, Ben. I'm really happy to be here and talk with you about my research and have the opportunity to meet all of you. I feel like it's a really exciting time to be in archaeology, and especially in the branch of archaeological science where I work. There are so many new tools available. There are so many exciting new things that we can learn. And so today what I wanted to do is just sort of show you some of the new technologies that have become recently available, what kind of potential they have, and how we're trying to, in my group, apply them to better understand aspects of the archaeological record, and in particular the zooarchaeological record, which have been very difficult to get at otherwise. And so today I've titled my talk The Archaeology of the Invisible, because a lot of what I'm going to talk about today is using molecular or microscopic techniques that are looking at things you can't see with the naked eye, and how this is really kind of presenting a new frontier in zooarchaeology. But first off, one thing I want to say is one of my favorite things about working in archaeological science and working in this field is that it is so collaborative. I work with many people in my work, and that's something I really value, is to apply some of these techniques. No one person has all of the skill sets that are required. So we work in teams, and we work with a lot of students and postdocs, students including undergrads, master's students, PhD students. And so I just show some of them here, and I find this to be one of the most rewarding aspects of my job. And I just want to acknowledge that a lot of the work that I'm going to show throughout this talk is not work I've done alone, but is work that I've done with students, with colleagues around the world. Another thing that I really like is, although these days, I did start off my career as a field archaeologist, but for more than a decade now, I've been working almost exclusively in the lab. But nevertheless, I still do get to sometimes go into the field, which I always really enjoy. And I feel like that's such an important component, is to be able to understand both the differences, the challenges, but also the strengths of having both fieldwork and lab work experiences. And so that's something also with my students, I try to make it so that all of them have both fieldwork and lab work experiences, because I feel like you can't really fully understand what we're trying to do here unless you've had both experiences. And another thing too is that one thing that's really important to me is training. And so I have a lot of visiting students that come and work in my lab and have worked in my lab over the years, to learn some of these new techniques. And if anyone's interested in this, I now have built up a new Zooms lab, Zooarchaeology by mass spectrometry, and I'm happy to have students come and train and learn moving forward. So first off, I just want to say, kind of frame some of the work I'm going to talk about today. There's many different ways to approach archaeology and to approach the past. And I think for many people, they think of it largely in terms of the monumental, in terms of the very large. And here you see, for example, a monumental burial mound from Mongolia. This is a Hirixur. This is located in Hovskol in northern Mongolia. And I think for a lot of people around the world, when they think of archaeology, they think of this. They think of archaeology as being very large, something that you can easily see that's conspicuous on the landscape. But in my work, I work with kind of a different end of the scale. I work with the archaeology of the very small, of the things that are you wouldn't see, you wouldn't notice. And here I show an image of a tooth, and you can see dental calculus on it, or calcified tooth tartar. And what I hope to do is convince you by the end of this talk that although archaeology can be at really large scales, there are also entire worlds to explore in these very small and humble materials, and that really this molecular record can provide really complementary and exciting new information to help us understand this much bigger context. And so I call this work that I've been doing the archaeology of the invisible, this molecular work, this microscopic work, all of these analyses that we perform that you can't actually see with the naked eye, that we have to approach through instrumentation and other things. But one of the benefits of looking at this very small scale is that in many ways, some of the very smallest archaeological remains preserve the longest. And so by focusing on the small, sometimes that extends the record by which the time depth and the breadth of the record that we can actually analyze. Now, as Ben mentioned, I have my kind of shoes in two places. So I'm a group leader at the Max Planck Institute for Evolutionary Anthropology, where I work closely with Johannes Krause. And most recently, I joined the faculty at Harvard where I am now an associate professor. And so I have labs in both places and do work in both locations. I'm currently in Boston right now, but I'm actually going to be traveling back to Germany for a year-long sabbatical later next month. So one of the things that I find really interesting and kind of drives a lot of the questions that I ask is trying to understand human behavior and how it's changed through time, and in particular as it relates to diet. You know, how did we go from eating out of environments like this? This is a photograph I took from the site of Gila-Nikitz in Oaxaca, Mexico. It's an early hunter-gatherer site. You know, how do we go from living in environments like this and foraging for our foods to eating out of environments like this, really within a comparatively short period of time? And what kind of impacts did these radical changes in the way that we eat, what kind of impacts have they had on us socially, biologically, culturally? So I'm very interested in many of these questions. And so today I'm going to kind of focus on how do we address some of these questions, and knowing how these dietary transitions take place, how changes in cuisine took place. How do we understand how food cultures develop, how they spread, how they influence each other, and the impacts these might have both, like I said, on our biology and our culture. Today, I'm going to focus mostly on our relationship with animals. And I'm going to be drawing on, you know, several different lines of evidence that we can use to help us understand the long relationship that we've had with animals and animal products, and how we can understand our changing diet with respect to animals. So first, what I want to do, though, is say, well, where do we get our information from? Like, how do we find out about this long prehistory of human interaction with animals? And of course, the most obvious one is by looking at animal remains. So using classical zooarchaeological techniques to examine animal remains that are present at sites. And there's a great deal that you can do with this. Everything, if you have diagnostic features, you can identify species. You can look at the parts of the animals that are being consumed. You can look at how the bones are being treated, whether marrow is being extracted or not, if certain bones are being curated or not, or used as tools. So there's a great deal of information you can learn about how people are managing their local environment, or perhaps managing animal populations, how they're cooking and how they're interacting with these animals through the physical remains of the animals themselves. However, in many cases, we have problems using these techniques because we don't always have diagnostic fragments. Sometimes the bones are heavily fragmented and they're not identifiable to species, or sometimes even to which element they might be. Or in other cases, such as fish, many fish can have very, very similar features, and it can be very hard to, even when you have well-preserved bones, to distinguish different fish species purely based on the morphology alone. So one of the techniques that's come around and has really been developed since around 2009 and originally developed by Mike Buckley is ZOOMS, or zooarchaeology by mass spectrometry, which takes advantage of the fact that collagen is the dominant protein within bones, and it preserves extremely well, and that's true of animal bones as well. And we can extract that collagen, and we can look at its sequences, and then we can make inferences based on the taxonomy. And while this doesn't tell us anything about which bones in terms of which elements being used, or maybe how the bone was processed, it can give us really good information about which species or which groups of species were being targeted. And so this uses mass spectrometry to investigate this. And ZOOMS has been incredibly powerful for doing large-scale screening of these small fragments of bones, which often dominate some assemblages, but for which we didn't have the ability to really have any taxonomic resolution. So this is really exciting, and this is something that we're expanding a lot in my lab to try to look at this at larger and larger scales. And in particular, it can be really useful at looking at questions like distinguishing sheep and goat, which can be very difficult to do morphologically, or distinguishing cattle and water buffalo, which again can also be really difficult. So ZOOMS is extremely useful in this regard, and I think it's really an expanding technique that I think is going to help address some of these long-standing problems we've had in zooarchaeology of distinguishing closely related taxa that have different societal implications, different economic implications. Another area where we can also expand using molecular techniques for zooarchaeological remains is using ancient DNA. And this is something I'm also working on. And this is some data that from a recent study that I'm working on with my postdoc, Taylor Hermes. And we're looking at sheep prehistoric sheep across Eurasia, and trying to understand if we can look at the spread of specific populations of sheep and how they might spread with people or mix or be traded along the steppe and mountain corridors. And so here you see what's shown here is a principal components analysis plot where you can see that African sheep, wild sheep, Chinese sheep, and kind of Central Asian sheep today are genetically distinct from one another. And this has to do with the fact that these populations have been separated in time. And then we can analyze archaeological sheep, and we can start to say, well, which ones do they resemble? Can we reconstruct a history of how these different groups of sheep came to be where they are? And can that inform us about ancient peoples and their behaviors and historical migrations or trade networks of ancient societies? Another place where we can actually get really rich information about diet comes from a rather unusual source, and this is the calcified dental plaque on the surface of your teeth. So here I've shown a tooth in cross section, and you can see a dental calculus deposit or a tooth tartar deposit. If you zoom in on it and look at it at high magnification, you'll see that it's composed almost entirely of bacterial cells. And that's because it's literally the dental plaque, this buildup of bacteria on your teeth that calcifies in situ during life. This process of dental plaque turning into tooth tartar or calculus is actually the only part of your body that fossilizes while you're still alive. And this happens when minerals in your saliva just spontaneously crystallize within the material. This is exciting because those minerals protect the DNA and the different biomolecules that are inside, and we can later dissolve that mineral away to release those biomolecules. So in addition to looking at the bacteria themselves, which in my own work I've done extensively looking at the oral microbiome, we also get aspects of food that become entrapped in these dental calculus, and we can learn a lot from it. So for example, we often get micro remains or tiny fragments of food that preserve. So this right here is a little bit of connective tissue from meat. Now we can't say what kind of meat, it's not diagnostic in that way, but it does appear. This is a phytolith or a piece of plant glass, and these are two examples of starch granules from different plant groups. So this one is consistent with plants in the tribe Triticea. So this is the tribe that includes wheat, and this belongs to the bean family or pea family. So we can start to make some inferences about diet just from some of the microscopic food remains that get entrapped in calculus. But one of the areas that I found most exciting is, in addition to these microscopic remains, we also get biomolecular preservation, and especially proteins preserved and trapped in these dental calculus deposits. So this is an example here of a milk protein that we have been able to identify from dental calculus. This is a chromatogram that you see here, just visualizing the protein, one of the peptides from the protein. But we can use this to then go back and reconstruct, in this case, that this individual was consuming milk, which is consistent with caprinae, sheep or goat. And there's other peptides that we can identify that can give us even higher species resolution. Another source of information about past diets, which is often overlooked, in part because it's not that common, but when we do find it, it can be enormously rich, is paleofeces. Paleofeces do not preserve in most places around the world. And I think we can all be grateful for the fact that generations of past feces have decomposed away. But when it does preserve, it can give us these really rich insights into the past. And it generally does only preserve in areas that are extremely arid or dry. So for example, in very dry caves in Mexico in the Southwest, we find paleofeces, but also we find paleofeces in places like salt mines. And so, for example, I've done work at the site of Cherabad in Iran, where we have really excellent preservation of DNA from paleofeces. And just to give you a sense of what paleofeces look like, here's a couple of examples. One thing that is immediately obvious is that when you look at ancient paleofeces, they are generally much more fibrous and contain a lot more plant material than what you would see typically today. And I'll talk a little bit more about the kinds of information, both microscopic and molecular, we can obtain from feces to help us understand the diets of ancient people. And then last, one other area that my group has been exploring is looking at residues, so residues in various types of pots. So we've been doing some work in Nepal, up in these transverse Himalayan valleys that served as long-standing trade routes. And in some of the tombs that we have been working in as part of a salvage archaeology project due to earthquake activity in the region, we have found vessels, bronze vessels, wooden vessels that have largely decomposed remains inside. It's not necessarily visually apparent what types of foods were once in these funerary offerings. Using molecular techniques, we can sometimes identify those. The preservation in these tombs is really extraordinary, in part because it's it's in the rain shadow in the Himalayas, so it's very, very dry and relatively cool. And here's an example of some of the preservation. So this wooden bowl is about 1,500 years old. In this case, we do get some visible preservation. These are rice husks, so we know that this bowl contained rice, but this gives you a sense of what it looks like. Other vessels like this, it's a bamboo cup. Inside, none of the kind of residue that remains is visibly identifiable. However, by using a combination of microscopic and molecular techniques, we can start to see and make inferences about what types of foods or beverages would have been contained. And so we're following up some early evidence using DNA and proteins that this cup might have held a fermented rice beverage. But all of these kind of techniques that I've mentioned to you today so far have really only very recently become possible. This archaeology of the invisible is something very, very recent. This ability to see this molecular ancient world is something that has only been possible due to very recent changes in technology, and largely due to this technology. We've probably heard about next generation sequencing, ancient DNA sequencing. Within the field of ancient DNA, it was the invention of next generation sequencing, a fundamental redesign of how we do DNA sequencing, that is allowing us to recover so much DNA from the archaeological record. It's a newer technique that allows us to really recover this highly degraded, damaged DNA that was previously not possible. And it's amazing to think that all of those analyses that we do are really done on this tiny little chip. So I realize it's about the size of a microscope slide, but from this one chip, we can gain over 1 billion DNA sequences. And this is how we're building up this understanding of the past. Another new tool that has really also revolutionized the way that we apply archaeological science is this here. So this is these new advanced mass spectrometry instruments that have much higher resolution and are much faster in their analyses. So here you see, for example, this is a Q exactive, a Thermo Fisher Q exactive, which is a really powerful mass spectrometry instrument. And this is what we use to analyze ancient proteins. And together, what these two new technological advancements, these new instruments allow us to do is effectively to see. They give us much greater focus and higher resolution to be able to see, to be able to detect these ancient biomolecules. And once you have that ability, as I said, many of them actually preserve over very long periods of time. So it's a matter of being able to detect them. But once you can, it opens up this entire world of possibility. So the work that we do, however, it is challenging. I mean, when we're dealing with biomolecules, they're everywhere. We are swimming in these invisible worlds of DNA. And so we have to prepare our samples. We have to work in very clean environments to ensure that we're not contaminating these ancient samples with DNA from the modern world. And so we work within these clean room facilities. And this is an example here. This is my technician, Raphael Stahl, working at the Max Planck Institute in our clean rooms there. And so we take a lot of precautions. It used to be that most people had never really seen someone dressed quite like this before, in a full body Tyvek suit, double gloved, masked, everything. But I think in the now post-COVID era, this is a much more common sight. However, unlike in COVID, where people wear this sort of protective equipment to protect themselves from a virus, here, Rafaela is wearing this protective clothing to keep her DNA inside and to not expose the ancient samples that we work with to her DNA. So I'm going to talk a little bit about some of these, a little bit more detail about how we recover some of these information, and I'll focus on a couple of different sources, and then we'll talk about some exciting new directions we can head in. I'm going to talk a little bit about first paleo feces and then dental calculus. So I just want to give you an example of what we can learn by analyzing paleo feces for food. I'm going to talk about three specific examples that you see here. They come from very different contexts. So the first is from ancient Mexico at a site called El Zape. I'm going to talk about some paleo feces that we analyzed from Cherbad from a salt mine in Iran, and also at the site of Durnberg, which is an Iron Age Celtic site at a salt mine in the Alps. Well, the first thing we have to do when we're analyzing paleo feces is determine to what degree how well preserved it is. Paleo feces, as I mentioned, doesn't usually preserve at all. But even if it does, the vast majority of paleo feces that have ever been found are actually quite decomposed when you move to the molecular level. Even if visibly they look well preserved, once you look at them at a molecular level, they've often undergone a kind of composting effect. But rarely we do find examples where that hasn't happened. Typically, that's only in places that are extraordinarily dry. And these three sites meet those characteristics. And so these three sites are some of the best preserved paleo feces that have ever been described. And we're currently writing this up for hopefully submission very soon. And one of the things we can do is we can look at the microbes that are inside to determine how closely related they are to, say, gut microbes versus, say, soil microbes. And here I'm just showing some examples of here when we do predictive analysis of the composition of the paleo feces. Green here represents bacteria known to be present in the human gut. You can see that across the board, these paleo feces are estimated to have very high proportions of gut bacteria. And so that's really good indicator. These are well-preserved feces and therefore are containing a real signal from the past. Within the feces, we also don't just obtain bacterial DNA, we also find human DNA from the person who deposited the feces. And here, what we did is we just analyzed the human DNA from each feces and plotted them against the genetic distribution of human populations today. And they make a lot of sense. So our sample from El Zape falls within the genetic distribution of indigenous peoples of the Americas. Our Durnberg sample falls within the distribution of European populations, and our Cherbad individual falls within the distribution of Central Asian and Caucasus populations, which is where we would expect this also to fall. So that's also a good indication that we have well-preserved feces, each one likely from a single individual, and we don't have serious problems with contamination. So now what we can do is kind of dig into, can we find some dietary DNA in here and what will it tell us? And this is work done by my student, Sandra Fagernes, who you see up in the right corner. And because these come from different regions, we would expect very different foods to be identified. And that's what we find, which is a good sanity check for us to make sure that our techniques are working. So for example, in the Mexican site at El Zape, the only dietary DNA that we could identify with confidence was maize. And that we would expect that. We know that maize overwhelmingly dominated the diet of the people that lived at this time in the region of El Zape. And so this makes sense that we have recovered maize. And there's also other evidence, paleobotanical evidence from the site that maize was present. So we have a good concordance of data there. At the site of Cherbad, we found evidence for this here is barley, but also for wheat and millet that were present, which again, we also know from the ethnobotanical record were present. This is a good confirmation also of this. What was really exciting, though, is when we moved to the Durnberg sample. Here, the DNA was really very, very well preserved. And so we can even say more about the diet. Here, we really found a wide variety of different foods that were consumed. This is from multiple individuals. This is not all in one fecal sample, but across several at the site. But we see people are consuming cattle, sheep, pigs. They're overwhelmingly consuming barley, but also a little bit of millet and wheat as well. We see that they're consuming apples and they're consuming strawberries and they're consuming wild berries, probably like a raspberry or a blackberry. Those are probably associated with fruit wines. And then we see also, which is really exciting, plants that were probably used as flavoring agents. So we see, for example, onion, different cruciferous vegetables, probably something like a cabbage, some beans, and other flavoring agents. So herbs, some herbs that would have been included. So this is exciting, and we're starting to get then a more complex understanding of some of these diets, but really at like an individual level. And again, we can compare this at this site. There's been really extensive paleoethnobotanical work that's been done, and we see a high concordance between those, which is exciting. However, what is nice is that in some cases, the genetic work can help to identify plants that were not seen using paleoethnobotany because of the ways, for example, the plant was processed or ground up or used as a flavoring agent. So it extends our ability to see some of the plants that are not as visible using paleoethnobotanical techniques. We also did some analysis looking at metabolites. These are small molecules, small chemicals that are present, and we looked to see whether or not we saw what types of small molecules are present in the feces. And again, we see things that we expect to see. So we see bilirubin and biliverdin. These are chemicals that are found within feces. They're actually what makes poop brown, if you were ever wondering why it's brown. And they're byproducts of hemoglobin breakdown. And so we found those in the paleofeces, again, confirming that the paleofeces itself is well preserved. We find things like primary and secondary bile acids, which are produced by the body during the digestion of food. And then we actually, we see copper stanol, which is another breakdown product, in this case of cholesterol. And then last we found enterodiol and enterolactone, which are microbial byproducts of plant digestion. So we can't say which plants these came from, but we can see that the gut microbiome is digesting plants. So we can put this all together and start to build up these understandings of diet. So as I mentioned, though, paleofeces are relatively uncommon in the archaeological record. We're usually limited to either dry caves or salt mines or other quite unusual environments. So in many ways, we can turn to another kind of bioarchive of diet, and that is dental calculus, which is much more ubiquitous in the archaeological record. And because it's calcified, it survives in many more contexts. So again, this is that same image you saw before, and I'm just going to zoom in so you can see it a little bit better. So here you can see a closer view. If we zoom in really far on the dental calculus, you can see that it has some really interesting properties. First, it's layered. You can see these individual structures here. That's because calculus grows in many ways like layers of an onion or tree rings, where you have a layer of dental plaque that forms, it calcifies, a new layer of dental calculus forms, it calcifies, and so forth. And it builds up over the course of an individual's lifetime. What's really important about this is it doesn't remodel. It just continues to grow. And so what we have here really on this woman's tooth is an ordered record of her life, her lived life. So we have the most interior layers formed earliest in her life, and the most exterior layers formed closely before death. And we can then look at these individual layers, and each layer then represents a discrete time period in her life. We can, as I said before, we can zoom in and take a much closer look at the specific bacteria that are present, but we can also decalcify this, and we can stain the cell walls of these bacteria and show that the bacteria are actually still fairly intact. They're so well preserved by these minerals that even when you remove the minerals, the cell walls don't fall apart. They're still intact, and they still absorb the dye and allow us, in this case, for example, to distinguish gram-negative and gram-positive bacteria, to look at some of the structure of this biofilm. But really incredibly, if we can use a dye here called Hooke's dye, which glows blue in the presence of DNA. So what you're seeing here is all of the DNA that's preserved within this particular sample of dental calculus. So dental calculus is amazing in this respect because it's the only known archaeological substrate that's so rich in DNA that you can actually see it under a microscope with the aid of these dyes. And then here, some of you, if you have a sharp eye, might have noticed this kind of strange thing here off into the left. This appears to be a phytolith, or a piece of plant glass that was incorporated at this point in her lifetime. It became embedded in the plaque and then calcified into the calculus. So again, you can also look spatially at the deposition of these different both biomolecules and microfossils to understand something about their experiences through life. I showed this image before, so we can dissolve the dental calculus, releasing different microfossils that then can be analyzed and studied to reveal information about diet. And we can also look at the molecular level. So really look at the specific DNA or proteins that are preserved. And right now, what I'm going to talk about is some of our work on proteins, because this is what we found to be most productive on reconstructing aspects of ancient diet from dental calculus. So we recently did a study in the Southern Levant, and we were looking at the types of foods that would have been consumed. And we were able to identify proteins from a wide variety of food sources, including sesame seeds, wheat, a protein that is found in ripening bananas, turmeric, and also soybean proteins. And this is helping us to understand the kind of origins of Levantine cuisine and when certain long distance trade goods entered into those food cultures. And we published this earlier last year. In this case, all of the remains that we identified were from plants. However, looking at other populations, we've been able to identify proteins from animals. So we did another study, this time on individuals who died in the Great Irish Famine during the 19th century. And we published this also a couple of years ago. Here again, we looked at dental calculus, and we took two different approaches. So we work with Monica Trump, who's a really skilled paleobotanist, and she identified a wide range of starch granules that were preserved in the calculus, giving us some insights in the types of gruels and the types of grains that were consumed. And my student, Ashley Scott, also looked at the proteins that were preserved. And in particular, we noticed that we found proteins consistent with chicken egg and also with milk. And we do know that milk was provisioned as one of the main foods in the workhouses that people lived in during this terrible period of famine. And so I want to kind of take a step back and say, so this is really interesting. Like, where can we go with this? What can this tell us? And I want to start with the example of milk, because I think milk is a great example of how using these molecular techniques can give us information that we weren't able to necessarily get using other techniques, and how this can really expand how we think about the past and how it ties into societal developments that have really major implications. But let's just start with milk. Why is milk interesting? Today, we produce globally like a truly extraordinary amount of milk. I'm not sure people really appreciate just how much milk is produced around the world. As of 2011, it was nearly 700,000 tons of milk was produced every day in the world. And that's a really hard number to wrap your head around. And since we're in the year of the Olympics, I thought I'd put it into a more familiar unit, and that's Olympic swimming pools. That's over 278,000 Olympic swimming pools worth of milk is generated every single year today. It's such an extraordinary number. It's enough that actually it's enough milk that every single person every single day could consume a quarter liter of it. And what's amazing, though, is that the vast majority of the world's population is actually lactose intolerant. So why are we producing such enormous amounts of milk? And where does this tradition and this history, this prehistory of milk consumption come from? And so this has long interested me as the history of dairying. In many ways, dairy foods are such as an incredibly strange food. They're really rich, they can be nutritious, but they're also fraught, they're difficult to digest. How did this come to be? When did dairy become something that was practiced on a global scale? So there's many reasons why milk is useful. Of course, milk is the food that mammals produce for their infants, and it is extremely rich and highly tailored for growing an infant. It's rich in sugars, and specifically the sugar lactose. It's rich in proteins, it's rich in fat, and milk is really perfectly balanced for a growing baby. The milk of different animals, though, is different across different species, and this is because different species have different nutritional needs. So, for example, Homo sapiens or human babies really have quite high lactose needs. And so human milk is quite high in lactose, actually very similar in amount to horse milk. However, for humans, we don't tolerate high amounts of protein. So the protein percentage of human milk is actually quite low, especially when you compare it to something like reindeer milk, where it's very, very high. And so these different proportions of fat, protein, and lactose are really tailored to the specific mammal and their growth needs. So humans need to grow a very large brain very quickly. Our body growth is slower, whereas other animals need to grow their bodies very quickly. So these nutrient profiles vary across these different species. But of course, while all mammals consume milk as infants, we as humans are quite unusual in that we continue to drink milk after infancy, and we drink the milk of other animals. And in that respect, we're quite unique in the world. It is really also very unusual that we continue to drink milk for so long. As I said before, most animals, they go through a weaning process and the milk is done at that point. They don't consume it, but yet many populations around the world continue to drink milk or consume milk products long into old age. And so, why is this? Why do we see these different patterns? Well, one of the reasons why other mammals don't continue to drink milk is because they lose the ability to digest the lactose in milk. Lactose is a type of sugar. It's called a disaccharide, which means it's composed of two sugars, one of glucose and one of galactose. And it's connected by this thing called a beta-glycosidic bond, which we'll talk about in a minute. This bond is hard to break, and it actually requires a very specific enzyme called lactase, which is encoded by the lactase gene. And lactase acts like a kind of molecular sugar, a scissors, to cut those two sugars apart. It's produced in the small intestine, and it breaks that beta-glycosidic bond between the two sugars. Our intestines cannot absorb these disaccharides. So instead, we must absorb, we can only absorb these simple sugars. And so we must break these sugars apart into their individual components for us to be able to absorb them into our body and derive nutritional benefit. This gene that controls, that produces lactase is on chromosome 2, but there's another region that's about 14,000 bases away that is actually the regulatory region, meaning it's the region that tells that gene to turn on or turn off. So as I mentioned before, this lactase is made in the small intestine, and during infancy, you make a lot of it in your small intestine. And so when lactose comes into your gut, the lactase breaks it down into the individual sugar molecules, and those sugar molecules are absorbed by your body, and that allows the infant to grow and derive energy from the lactose sugar. However, if you do not make lactase in your small intestine, the lactose just continues to flow by. It can't be absorbed. And it continues to flow through your digestive tract until it hits your colon, which is where you have billions and billions, in fact, trillions of gut bacteria that reside there that would be very happy to eat that lactose for you. The only problem is that when they do, they release as byproducts hydrogen gas and other irritants that are responsible for creating the symptoms of lactose intolerance. So it's long been known that, as I said before, this regulatory region, which is in a gene called MCM6, controls the expression of lactase. So what we see is that during infancy, the regulatory region turns it on high, so you produce a lot of lactase. But as you get older and older, the amount of lactase that you produce decreases until finally you stop producing it. And this is actually the natural state of all mammals. All mammals undergo this process, and it's part of the normal weaning process. What's interesting, though, it's been, as I said before, is of course, but humans continue, some human populations continue to drink milk. So what's going on here? What's allowing it? Now, it's been known for some time that this region of this regulatory region actually has a number of mutations. And we know that, for example, if you have a mutation here or here, then you just produce lactase as if you were an infant for your whole life. And these, we know that these mutations here cause the same phenotype. And these ones here are common in European populations, Anatolian populations, and Iranian populations, and also in South Asian populations. These ones here that I showed are common in populations in the Arabian Peninsula, especially amongst Bedouins. And we know that these ones marked here in purple are common in, are seen in Tibetan populations, although a lot less is known about these specific mutations. And these ones here shown in blue, and there's one that's actually in a slightly different region, are found in East African populations. All of these mutations have a very similar effect, and that is that it causes lactase to be produced for the entire lifespan of the individual instead of only during infancy. And so for a long time, it was thought, well, this is the explanation. We now know why some populations drink milk and why others don't. It must be that these mutations kind of arose by chance and then were selected for in these populations as they developed dairy technology or as they were developing pastoralism. And so it was argued that this probably developed quite early in ancient Neolithic farming populations, and then spread with those populations as they moved, for example, from the Near East into Europe. And so it was thought for a long time that those first farmers of Europe probably brought this special genetic trait with them that allowed them to digest milk, and that explains the high degree of lactase persistence in Europe. The problem, however, came with this really kind of like bombshell paper published in 2007 by Joachim Berger and colleagues, where they performed genetic analysis on these early Neolithic farmers in Europe and showed none of them have these mutations. In fact, they all would have lost the ability to produce lactase as adults. And subsequent work since then over the past decade has confirmed this over and over and over again. In fact, we find that Neolithic populations across the board do not have these mutations. And of course, one thing that was never explained is that there are populations today around the world who have a strongly dairy-based economy who don't have these mutations even today. And a good example of this would be Mongolia, and that just remains unexplained. If we take a perspective on Eurasia as a whole and look at where different dairying populations are, you can see that there's many, many different populations that have incorporated various aspects of dairying into their food cultures. I'm just going to talk briefly about this Mongolian example. Mongolia is quite special. They milk more species of animals than any other country. So 7 different species of animals are milked in Mongolia: horses, camels, reindeer, yak, cattle, sheep, and goat. And they're produced into a wide variety of fermented products. And the amount of dairy consumption is quite high. It makes up a really relatively high proportion of the diet. This is data from 2012 and 2013. And prior to the Soviet period, it was even higher. A lot of wheat was introduced in the Soviet period, but before that, the amounts of milk in the diet were even higher. But even the carbohydrate input from these dairy products is still quite high, and this is almost entirely coming in as lactose. So how is this possible? Especially because we know that from other studies that this region largely lacks these lactase persistence alleles. So how are they able to digest this dairy in their diet in the absence of having these special mutations? And why would you perform dairying on the steppe in this location? What would be the motivation for doing it? And if we study this Mongolian example, could this help us understand the origins of dairying in general and what might have motivated people to develop this rather unusual food? When the steppe in particular, dairying is really a solution to this environment. One of the problems of the steppe is that although it's a rich grassland, humans cannot digest grass. And so there's very little that human populations can eat in an environment like this. However, if you are managing animals, especially lactating animals that are either ruminants or equids, they can convert that grass into tissues and fluids that then become nutritious for humans, such as milk and meat. And so dairying becomes a way of converting these grasses into human consumable foods. And they do this through the action also of involving microbes to ferment these dairy products and enable digestion. But we have a lot of questions about the prehistory of this. You know, how and when did dairy pastoralism spread across the Eurasian steppe? And was it associated with migration? How did it spread? Who brought it there, and in what way? And which animals were the focus of this early dairying? And how are microbes connected to this? And so these are some of the questions that were really driving our research, and we turned to the archaeological record to try to understand it. Here's just kind of a bird's eye view of Eurasia here. And what do we know about the history of dairying? So we do know that the earliest evidence found to date of dairy production comes from about 7,000 BC in Anatolia. And this comes from analysis of pottery residues conducted by Richard Evershed and colleagues. We know that fairly quickly it spreads to Europe, and we see there's evidence of dairy fats in what have been called cheese strainers or these perforated pottery vessels. And we also know that fairly early it also gets incorporated into African economies and is present by at least 4,000 BC. And we know that there's some evidence that horses might have been milked at around 3,500 BC at a site called Botai, although that's still quite controversial. And we know, though, that likely a lot of the spread of these dairy technologies might have been facilitated by populations living in the North Caucasus around 3,300, 3,000 BC, something in that, because we see that these populations genetically, their descendants end up widely spread across Eurasia. And we think they may have been some of the people that spread these dairy technologies because we see their descendants in the region of the Altai Mountains by about 3,000 BC. And we know that other populations from the same region had a huge impact on Europe slightly later. And then even then we have other migrations that come from populations after they've gone to Europe, migrating back into the Altai Mountain region. And so we see a huge amount of movement across this steppe zone. And this might have been a major corridor of transmitting these pastoralist technologies and specifically dairying technologies. And we also know from direct evidence of entire, preserved cheese necklaces and kefir from the Tarim Basin that it was present in the Tarim Basin by at least 1800 BC. So, but how exactly was this going on, and what's going on in Mongolia, which hadn't been studied until, or dairying specifically hadn't been studied until we started working there. And so we started working in Mongolia. We partnered with Erdene Myagmar at the National University of Mongolia, and we tried to do a really wide sampling across time and space in Mongolia to try to determine when and where dairying arose and in what kind of cultural context. We looked at a number of different groups, different mortuary types, and different time periods and locations. And we analyzed teeth, both the teeth for the human DNA and also for dental calculus. And we extracted DNA and proteins from both of those and processed those samples in our lab. And we used the tools of both proteomics and genomics to better understand this history. Now, milk itself has many proteins in it. These are just some of them. And so we wanted to know if we could find any of these. And one in particular called beta-lactoglobulin is one that we find preserves very well in calculus. And what's useful about it is the sequence of it is different between different species. And so it allows us to reconstruct which type of milk, which livestock milk might have been consumed. And so when analyzing this particular context, we found really rich evidence for dairy consumption. We identified many, many peptides associated with different milks. And throughout the data set, we were able to identify the milks of goat, sheep, cattle, camel, and horses from a variety of different proteins. And in particular, we saw that it was structured, however, through time. So the earliest milk that we see is from sheep. And then we also see fairly early cattle and goat. Much later, during the late Bronze Age, we see the first, and the cattle, sheep, and goat comes around 3000 BC in association with the Afanasevo culture. We see that much later during the late Bronze Age, we see the first evidence of horse milk, after which that spreads very wide. And then we see the first evidence of camel milk only during the Mongol Empire. We also, looking at genetics, were able to model how this population had changed through time through interacting with neighboring populations. I'm not going to go through this too much, but this is a brilliant work by Chunwan Zheng, who's a population geneticist, and looking at the dynamics of the population in Mongolia over about 6,000 years. So I'm not going to talk about this very much, but just really quickly, one of the things I want to say is throughout this whole period, over 6,000 years, one thing that's really fascinating is despite being in contact with many neighboring populations, including populations coming from Central Asia and from the Caucasus region, we never see lactate persistence ever rise. We can see that it gets introduced by subsequent populations later on, but it's never selected for in Mongolia, despite the fact that over this entire period, people are consuming milk. So this is a really fascinating and puzzling case, a kind of enigma, as it were, in the story of dairying. So we published these in a series of 3 articles just a couple of years ago, and I'm happy to answer questions about those later. But we're starting to put some of the pieces together and to try to understand. So now we can put a date on when dairying begins in Mongolia. It begins at 3000 BC. We can see that it's associated with Afanazevo cultures. And we know the Afanazevo are closely genetically related to people from the Caucasus. And so we turned our attention to the Caucasus and asked, well, when did dairying begin there? So just kind of taking a step back earlier in time here and mentioning again that sort of this 3300, 3000 BC, something around here seems to be important. We wanted to understand the context of how dairying, what the dairy economy was here and how dairying got started because of its impacts on these neighboring regions. So I just have a few slides I'm going to show here. This is brand new work, and I can't show all the results just yet. We're preparing this for submission now, but you can see we've now sampled quite a few sites throughout the North and South Caucasus, as well as neighboring regions in the Trans-Urals and the Don Volga region. We have fairly good temporal spread from about 6000 BC all the way up to the first millennium CE. And we are finding really amazing preservation of milk protein. So stay tuned, I'll tell you some more, but we think we now understand some of the internal dynamics of how dairying is really developing here from small scale dairy production to fully blown mobile pastoralism with dairy, and how this might affect later migrations and the spread of this technology. Uh-oh, I think I froze. Okay, there we go. Froze for a second. Just in the last couple of minutes, I just wanted to mention a couple of things. So paired with this archaeological work, we're also doing work in ethnography and microbiology to better understand how people use and produce dairy products today. So we've been doing a lot of work in Mongolia. We've been working with different herders and learning about how they make different dairy products and learning about how they care for the microbes that are used to produce them. And these are just a couple of examples here. If you're wondering what's happening in this far right panel, this is Gerol. She was demonstrating how to distill vodka from yogurt. And so that's what she's doing here. She's finished her distillation and she's cleaning the still. But just as an example of some of the variety of dairy products that are made today in Mongolia. We started a project called the Heirloom Microbes Project to try to understand how dairy is produced and what sorts of microbes are used in these different processes. We've been mapping the different types of dairy products, how some dairy, you go from milk to different forms of dairy, and then how they are used. And we're looking at the different microbes that are present in each, because ultimately we'd love to be able to also trace these microbes back in the past, which might give us clues not only to which livestock species these are being milked, but what products they might be making with those. The government makes us import them in biohazard bags. So this is just some of the examples of this, bringing back these different dairy samples that were collected in Mongolia. It's been really challenging because foot and mouth disease is endemic in Mongolia. So we have to go through really extensive testing before we can bring back our dairy products for testing. But we've now have many of these in the lab and been able to characterize them both nutritionally and also for the microbes that they have. We've identified a wide variety of microbes that are present. We're starting to explore what their functions are and some of their properties. And then last but not least, another aspect of our project is now we're trying to understand, going back to this question of how are people digesting these dairy products and digesting this lactose if they don't have these lactase persistence mutations that allow them to produce lactase. As I mentioned before, it's really the action of the bacteria in the colon which determine the lactose intolerance symptoms. And so we're now conducting a large-scale gut microbiome study in Mongolia to try to determine if they have specialized adaptations in their gut microbiome to be able to digest milk. And so with that, I just want to say thank you for your attention. I'd be really happy to answer any questions you might have about the new techniques that we have now available in biomolecular zooarchaeology. And just as a shout out for anyone who might be interested in, Ben mentioned this earlier, I have a coloring book project called Archaeological Adventures in Archaeological Science. We now have it available in 33 translations and in many languages around the world. And if you'd like to learn more, I'd be happy to talk to you about it. Or if you speak a language we don't already have and would like to help us make that coloring book available in a language where you live or work, we would love to partner with you. So thank you everyone for your attention, and to everyone who contributed to the research that I presented today.
Thank you very much for this fascinating lecture. That was great to see like the